Starting with this worksheet, you have to know these notes. You have to know what is transgenesis. It's the process by which a gene is transferred from one species to another and is expressed in the later. Transgenic is an organism that carries and expresses the gene. Let's start with question number one: Arctic place and antigen gene. If a species called the Arctic place is equipped with a valuable gene, antigen gene, which is responsible for the synthesis of an antifreeze protein that enables them to resist very low temperature. So this fish, they have an antigen gene. This gene is responsible for the production of antifreeze protein that enables them to resist very low temperatures. On the other hand, salmons, they die when the temperature decreases below 0 degrees Celsius. For that, researchers, they wanted to equip salmons with a severe antifreeze gene. So they have to transfer the gene from the Arctic places, the fish, toward the salmon. Okay? As shown is this, DNA is extracted from a liver cell, and then antigen gene is transferred to multiplication of bacterium, and then microscopic needle injection of humerus gene into the salmon egg, and then was injected into the salmon, which was mutant. Write a short text. We're going to describe this by a short text. And I'm going to define the term mutant. You describe the obtained salmon is not proper in this case. Propose another term. Another term for mutant can be used transgenic or genetically modified organism, GMO. So genetically modified organism, or it's transgenic. What new characteristic will the mutant salmon acquire? It acquires the ability to resist very low temperatures, since it has this valuable gene اللي هو اسمه the antigen gene. Question number two, transgenic sugar beet. A viral disease, isomania, of the sugar beet has been spread throughout Europe since 1950s. When a culture of sugar beet is contaminated with the yield loss may reach 80%. Also, the sugar content of beet passes from 18% to less than 10%. Thanks to biotechnology and the progress of genetics, the gene responsible for the synthesis of protein envelope of the Rhizomania virus was isolated and then introduced in a stable manner into the genetic material of the sugar beet. The objective is to enhance sugar beets to produce small amounts of viral envelope protein, which provokes a defense. So what is the objective of this? Objective is to enhance sugar beet cells to produce small amounts of viral envelope protein, which provokes a defense response against this virus. This response is effective by inhibiting any attack of the plant by the same virus. Like all viruses, that of Rhizomania has the ability to mutate very easily. Thus, this defense becomes quickly useless. Pick out according to the text. The name of the disease attacking the sugar beet. What is the name of the disease? It's called Rhizomania. There's the consequences on the beet plant. What will happen for the beet plant? You have to pick it out from the text. Throw out by referring to the text the steps of the technique. All of them, they are picked out. Explain why the manipulated sugar beet is GMO, genetically modified organism, because we introduced another gene that is responsible, or it's responsible for the synthesis of the envelope protein of the Rhizomania viruses, and so to protect them. Indicate by referring to the text the advantages and the inconvenience of this biotechnology. So let's move to the solution of this worksheet to talk about it and to be in front of you. Okay? And this solution is uploaded on the drive. You can see the solution later on. Okay, guys? So first question, they said, describe DNA was extracted from the liver cell of the Arctic place fish. After the antigen gene is isolated, it was introduced into bacteria. So the transferred bacterium, they multiply, and they consequently, the antigen gene also multiply. With a microscopic needle, many genes were injected into the salmon's egg, and a mutant salmon was obtained. Transgenic salmon was the result because the mutant salmon comes from the egg which has been subjected to transplantation of a gene from another species. The mutant salmon will be able to face cold or to resist cold and to live under low temperature. What is the name of the disease? It's Rhizomania. What's the consequences? The yield loss may reach about 80%, meaning that 80% of the sugar beet will be lost. The sugar content of beet passed from 18% to less than 10. What is the importance? Isolation of the gene responsible for the synthesis of the protein envelope of Leishmania virus, introduction of the gene into genetic material, reduction of small quantities of viral. Why is the beet considered as GMO, genetically modified organism? Pay attention to this because it receives a foreign gene. It receives a gene from another species and integrated into the genome, into the DNA, which gives her a new property to defend against Rhizomania. Advantage: the modified sugar beet has a defense system for inhibiting the attacking by Rhizomania virus, while indeficiency or inconvenience for this is the defense response induced by this method could be quickly useless because Rhizomania has the ability to change itself, to mutate itself. So this is our new chapter for the next week about mutation and what the meaning of mutation and what happens during